Good afternoon, everyone. Thanks for joining this webinar. We'll start in a few minutes just to give other people time to connect. Thanks for your patience. Hello, everyone. I would like to welcome you to today's webinar, Ancient DNA Panel Improves the Protocol for Highly Degraded Samples, the case of mother-child burial in Alwise. My name is Gemma Garcia, and I am the EMEA Regional Marketing Manager at Twist Bioscience. I would like first to give you some housekeeping guidelines. All lines will be muted during the webinar. If you have any question, please submit it in the Q&A box rather than in the chat box, and we will answer the questions after the presentation. If you share your name, we will be able to get back to you after the webinar if your questions have not been answered. Following the presentation, if you have 1 minute to take a brief survey, we would appreciate your feedback. We are recording this webinar. Now, I want to introduce you to our speaker for today. Nicoletta Zedda is postdoc researcher at the Department of Environmental and Prevention Sciences in the University of Ferrara in Italy. I am glad to announce that the university will have soon an ancient DNA laboratory. She has a strong research experience in biological, molecular, and archaeoanthropology, as well as in osteology and medical anthropology. After completing her bachelor's degree in natural sciences at the University of Cagliari in Italy, she obtained a master in quaternary, prehistory, and archaeology in 2016 at the University of Ferrara, Italy. And she did the PhD in 2020 in physical anthropology, also in Ferrara, with a project on influence of health status on plague mortality, studying a sample of plague victims from 1630. In 2021, she did a postdoc project to improve the protocol extracting ancient DNA from highly degraded samples, with a collaboration between University of Ferrara and Ancient DNA Laboratory at the University of Mainz in Germany. It was during this period when she tested the Twist Bioscience panel for the first time. In this presentation, she will show you some of the results obtained on the protocol, where that she used to study recently published in scientific reports. Nicoletta, all yours.
Thank you, Gemma, for your presentation. So, as Gemma already told you, I am a physical anthropologist at first, and it's only in 2021 that I ventured into the world of ancient DNA, so to say, with a Go4It project, a project funded by the Conference of Rectors of Italian University. And my project had the aim to establish a protocol for the ancient DNA analysis of highly degraded archaeological skeletons. And this project was a collaboration between University of Ferrara in Italy and the Johannes Gutenberg University of Mainz in Germany. Particularly, I was with the paleogenetic group of the University of Mainz that I just want to briefly present. I mean, the paleogenetic group is led by Professor Joachim Burger, and has many members, actually, master student, PhD student, postdocs. These are just a few of the members, but they are the ones that were directly involved in my research. So the archaeologist Maxime Bramy and the postdoc bioinformatician Jens Flucher and Laura Winkelbach, that is a research lab technician. So before presenting the results of my research for the protocol that I use in my study, why is ancient DNA and for a certain reason, also forensic DNA, challenging sample. Many of you probably already know, but ancient DNA is degraded, damaged, and fragmented DNA. And we have very, very low amounts of it. And also it has going on some post-mortem based modification, for example, hydrolytic lesions. And of course, it's prone to environmental contamination. Of course, how much the DNA is preserved, how much it's preserved in the modern day depends on the environmental condition that can help or not in preserving DNA. So, for example, of course, in arid, cold places, DNA is very well preserved, but that's not something that we can act upon. What we can do is trying to limit the modern environmental contamination, and that should be done starting from the sampling of the DNA. I would like to say starting from the archaeological excavation is possible. So from as soon as possible as the material is recovered, it should be try to limit the modern environmental contamination. So the protocol that I want to present starts from the sampling itself. So first of all, what do we sample for a recovery ancient DNA? We can find ancient DNA from a multitude of sources, from mummified tissue to dental calculus to the sediment itself. There are many studies in recent years that are trying to recover DNA, even human DNA, from the sediment. But the main source of human DNA, the best source of human DNA, of course, are bones and teeth. In fact, collagen, that is the organic part of the bone that is protected from the mineral part that form the bone, or dentin in case of teeth. The collagen is preserved in this way more for longer. And so from the skeleton, potentially all part of the skeleton, in particular compact bone, can be a source of DNA. But more and more studies, and here in the slide you can see a quote of a recent study of 2020, showed many studies show that there are some parts of the skeleton that are the best part in preserving DNA. And the best of all, the golden rule is to use petrous bone, so the inner ear bone, because inside of the petrous bone there is a very dense bone part that preserves DNA really well. Then, of course, there are teeth. Teeth are good as well, but less than petrous bone, and because, of course, it's protected from enamel and from being inside the alveolar bone of the skull. Of course, they are. I say they are less good than petrous bone, but that depends on what kind of studies one wants to pursue. I mean, in case it's not in endogenous human DNA that one wants to study, for example, but want to study not only that, but also hoping to find the DNA of pathogens, for example, pathogens that affected the individual in the past. The best part to sample in this case are actually teeth, because the dental pulp inside the teeth is highly supplied with blood, and therefore it's more possible to find traces of pathogens that were circulating in the blood of the individual than not petrous bone, for example. And in this particular paper that I quote here, they did some comparison on the different parts of the skeleton, and they found out that another good part for preserving DNA is actually the thallus bone. That was surprising to me at first, because it's not very compact bone, it's more cancellous bone, the one inside the thallus, but actually I tested it, and it's I mean, it's not as good result as petrous bone, but it did have good results. So in cases of in cases the cranial bones are not preserved for some reasons, because it happens, then thallus bone is a good substitute. So what we sample, sampling should be done in a way as I was telling before, to minimize the risk of contamination. So using gloves, hairnets, mask, cleaning and sterilizing with soap and bleach all the equipment and surfaces between each samples to avoid also cross-contamination. So in this picture, for example, I'm showing a sampling of a petrus bone. In this case, I have used this particular cylinder drill point of 1 centimeter of diameter. You can see in the right lower part. And this allows me to take off the very dense part of the petrus bone without breaking anything, without breaking the skull, taking only what I need, and then take this part inside the ancient laboratory lab, the ancient DNA laboratory lab, and clean it really well, removing all the remaining dirt, and this really limited the environmental contamination. So once the sample is taken, of course, it is transported to an ancient DNA lab. The lab should be, probably you already know, but an ancient DNA lab should be physically separate from other molecular biology laboratory to avoid contamination from modern amplified DNA. So the people who work inside the lab, in ancient DNA lab, should respect strict measures to avoid bringing contamination of modern DNA from the outside to the inside. And so changing clothes, wearing suits, gloves, helmets, hairnets, mask. So all kind of precautions to avoid bringing DNA, modern DNA inside the lab. And of course, there are all measures of cleaning all the surface and the equipment between the samples to avoid, again, cross-contamination. And moreover, all the rooms in the lab are daily exposed to UV irradiation to avoid DNA deposits. As you can see in the picture in the lower part, for example, that's the ancient DNA lab of Mainz. That during the night, it's exposed, all the rooms and surfaces are exposed to UV irradiation. So what do we do with our sample that we have taken, that our piece of bone that we have taken, that we have sampled, we first document it and decontaminate with the UV irradiation itself. Then we clean it from any remaining dirt. In Mainz, we also use sandblaster to clean really dirty samples. And I find that being really accurate in this phase, even if it slows down the process a little bit, can really change how much endogenous DNA is recovered. So cleaning really well the piece of bone is really important. And at this point, then the bone pieces are pulverized and are ready to be extracted for extraction. So I'm showing here the extraction protocol that I used in Mainz. Of course, we can a better description can be found in the paper. I wrote it in the slide if you are interested, but they are mainly 4 steps. So there is a pyrolysis with 1 milliliter of aDNA at room temperature for 10 minutes. And we use between 0.10 to 0.15 grams of bone powder. Afterwards, there is the real lysis. The lysis, it lasts for 48 hours, 37 degrees Celsius degrees on rocking shakers, so to avoid deposition of the bone of the powder. With 1.8 milliliter of buffer. The buffer is composed from aDNA, 3-HCl, and laurocylphosine, and proteinase K. After 48 hours, the supernatants are transferred to an amicon filter, and that's a really important step, actually, because through the amicon not only allows to concentrate the DNA, but also to eliminate all the smaller, not useful for us fragments of DNA. And so the Amicon filter is of 30,000 Dalton, and the DNA is washed. This filter is washed twice with 1x3 DTA, and afterwards the concentrated DNA is purified and eluted using the Kiagen mini-elute kit. So that's the main steps of the extraction protocol that we perfectionate. The library steps are actually not so innovative. I mean, they are based on the description on the library protocol reported in Kirchen et al. Of 2012, with some small modification. I mean, for example, the main one is the use of 3 microliter of filling product and 20 of acuprime supermix for each PCR parallel. And of course, the indexing, I use the indexing sequencing from the Nextera XT index kit of Illumina. PCR cycles, they are done using manufacturer instruction, but with a 60 degrees Celsius annealing for 30 seconds during each cycle. And we usually did between 10 to 14 cycles of PCR. Usually they were all between 10 and 11, 12, rarely for more than 12. What I usually did before doing the main sequencing was doing a first shallow sequencing, shotgun sequencing, to evaluate really shallow, to evaluate the endogenous content. And usually the process, the protocol said that if the DNA content, endogenous content, was really, really low, unless the sample was really important, we would not process, it would not be processed over, because there was not enough DNA to, I mean, think about if you have only 1% DNA content, endogenous content, how many complete flow cells, for example, of a Novasec, that is what I usually sequencing on during my research in mines, how many flow cells would be needed to have a 1x coverage of that genome. So it would be really expensive. So what I did was to try to test this new, at the time, 2021, the new target enrichment panel. There was the twist capture panel for ancient DNA. It's a panel that captures 1.4 million SNPs that include the 1240,000 SNPs panel, classical panel for ancient DNA, plus some new unique content. And it also has the characteristic that it's this new probe design with a native base pair that target the third allele and so eliminate bias, and also has only one round of hybridization. So I was not the first one to test it, of course. Other researchers had already tested the panel, and in the next year, on the 2022, Roland and colleagues published already the paper where it was described all the protocol that they used, and it proved the enrichment powers of the dates, of the twist dates. But we wanted to test it as well, and also test its sensitivity, so to say. So I performed 2 experiments with different samples with very low endogenous content, so samples that we already did the shallow sequencing, and we tested that it was really low endogenous content, between 1 and 10% of endogenous content. And I did also these 2 experiments, one using 2 independent libraries from 1 extract for each sample, and one experiment using 4 libraries from 2 different extracts for each sample. So the aim was to see if incrementing the complexity of the initial sample would have better results in the sense of how many SNPs were covered, how many reads were on target. We used the same protocol that is reported in Roland et al. In 2022 with 2 small modifications. So the double index library were pooled and concentrated to obtain 1,500 nanograms instead of one library with 1,000 nanograms. So we did 2 libraries for 1,500, and we used only 12 cycles at maximum, and not 23 that they used in the 12 PCR cycles. Sorry, I didn't wrote it, but 12 PCR cycle instead of 23 PCR cycle that they use in their protocol. So first of all, we noticed that the target enrichment, of course, increased the general endogenous content of all the samples. That was expected. And the second experiment gave better results, but not for all the samples. So it worked better with the best with those samples that had an initial endogenous content that was higher, of course. That was not surprising. What was interesting is that the on-target SNPs, in this graph, I'm only showing the 1240K SNPs only. The on-target SNP didn't change so much between the 2 experiments, showing that even with low complexity, so even with the experiment with only 2 libraries per samples, we already reached almost all the SNPs that could be captured. And what changes is the depth of coverage, of course. So with the second experiment, the depth of coverage was much higher than the first one, of course. So with the same sequencing, the same amount of sequencing, we had a much better depth using more libraries than using only 2. So in conclusion, what I wanted for those 2 experiments that I did, what we see that increasing the complexity of enriched pool library leads to an increase in depth of coverage, but the number of captured SNPs is only slightly positively affected. Because the reagent, the capture reagent performed with high sensitivity for all the samples, even with low complex pooled libraries. And that's really good, of course. So I did not only did that in my 18 months in Germany, of course, I also used this capture in other projects. In particular, the project on the prehistory of Luxembourg. It's a project that was led by the archaeologist Maxime Bramy, and it was funded by the INRA of Luxembourg. And we analyzed the genome of 33 samples from Luxembourg, from the prehistory of Luxembourg, from Neolithic to Iron Age. There are some preliminary results are already published in the project gallery of the Antiquity Journal, but more will be published in the near future, hopefully. What I want to show you today is one particular case that was already published last year, is the case of the mother-child grave of Alwise. This grave is from the Belbica culture and was discovered around 2000 during some excavation of Alwise in Luxembourg for reconstruction of a highway. So it was like an emergency excavation. This burial is dated to the 3rd millennium BCE. As some of you may know, or maybe not, but I will tell you anyway, at the time of the 3rd millennium BCE, societies predominantly buried the dead in single graves. So there is, like in the third millennium, this widespread shift from collective burials that was more common in Neolithics to individual inhumation. So for such societies, this shared inhumation with more than one individual represents an exception. So archaeology consider this adult child burial as special or deviant burials. The grave of Alwise, these two skeletons are a skeleton of a woman, a young woman of 25 to 35 years old, and a child around 3 or 4 years of age. They are buried at the same time together. The head of the child is resting on the right hand of the woman, and the two individuals are facing each other. So this position of the bodies and the presence of some particular grave goods, and in particular the presence of fossil around the skeletons, made the archaeologists of the groups thinks about their resemblance to another grave, that is this one, the grave of Dunstable Down in England, that was discovered in the late 800s. This is also known as the echinoid burial, as well belonging to the bell beaker culture. So we decided that it would be interesting to confront these two burials, to try to answer the question on why were they buried together, the adult and the child. If there was some relationship, familiar relationship between the adult and the child. To check also if check the closeness regarding the genetic composition between the couple, the woman, the adult and child in England and the one in Luxembourg. So, for example, in particular, to see, for example, if the British individuals had the same amount of step ancestry compared to the Luxembourg couple. So we did extract DNA from all 4 individuals, and we did this comparison, and the results are published in the paper in scientific reports last year. Unfortunately, and that's why I'm talking about it today, the Alwise grave was very bad preserved. In particular, the woman, the adult woman, had really low endogenous content. We are talking about 1% endogenous content of all the DNA extracted. So we decided to use the twist panel to target enrich the DNA. And this allowed us not only to place the sample on a PCA plot, as you can see in the picture, so they are all belonging to with the other samples from the bell beaker culture, so confirming they are belonging to that particular culture, and also from the admixture graph on the right, you can see that all 4 individuals have had a high component of step ancestry, so they are all belonging to this, how can I say, to this migration that came from the steppes in Europe and distributed to all Europe and had and established there. So not only was possible to do some populational analysis, but also what was more interesting was the kinship analysis that we could do. In particular, from the woman and the child, we could find out that the woman was indeed biologically a woman and the child was a male boy. We did these are the programs that we use for the kinship analysis, of course. So, of course, this kind of analysis of kinship would be impossible to do without the target enrichment, but that was what allows us to understand that they are indeed mother and son. I mean, that's not so surprising here. There's no big surprise here, actually. What was interesting about these 2 individuals was that from the archaeological point of view, in fact, it seems that we know that at that time, the orientation of the bodies in the graves was very codified. They have a very strict rule, with some, of course, with some regional and temporal differences. So the orientation of the body in Alwise follows the male orientation. So the boy is positioned correctly, if you want, while the woman is not positioned following the rules of the female orientation, because it was positioned to face the child. So it seems that the male boy, in a certain sense, was, we can possibly say, more important, is the one that gave the orientation to the grave, while more important than the mother, at least for whoever buried them. So the other individual, the other couple actually was a bit more interesting. They had a bit more, not so a simple relationship. They were not mother and child. It was an adult woman of 25 years of age, and a child, a girl child around 6 years of age. And they were actually paternal aunt niece. So it was more interesting, a bit more complicated relationship. Again, on the paternal side, so it seems like that's the suggestion that the archaeologists did, that this Belgica culture had more of a patriarchal relationships, more important. The patriarchal line, the paternal line was more important. Of course, these are just suggestions. Of course, another important thing that we could do is the reconstruction of the phenotype, so the coloring of the woman and the child of Alwise. The woman, for example, had blonde hair and blue eyes. The child was more of a brunette, and it was this information were used to have an artistic depiction of the two bodies. So the question of why were they buried together? There are many cases of adult, not many, but there are some cases of adult child burial, and sometimes it's really clear why they are buried together. For example, in cases of violence, we have found some other individual that were that shows very clear sign of violence, but in this case, it was not so clear. So we couldn't find any signs of violence. Archaeologists that proposed that one reason could be sacrifice, so that maybe the child was sacrificed because the mother died or something. But at least for the case of Alwise, this seems not probable. Still, it cannot be completely excluded. Another possibility, of course, is infectious disease. So unfortunately, we could not find any sign of infection or pathology that was clear in the skeleton, but there are many cases of infectious disease that are so fast that cannot leave any sign on the skeleton. Let's think about the case of the plague. I mean, I have worked on plague before, but now, in the last year, there are many cases of Yersinia pestis being found in Neolithic and Bronze Age samples. So it cannot be excluded, but we could not prove it here. Finally, I have a last thing that I want to show you, and it's this new project that I'm currently working on with the University of Ferrara. The PI is Professor Barbara Bramanti, and it's funded by the European Union and the Italian Ministry of University and Research. And it's a new project. It's called Shoveling into the North Italian Bronze Age with an integrative approach to unearth the evolutionary origin of non-metric dental trait. So it's a project we are analyzing these 23 samples from the Terra Mare culture. It's from the Middle to Final Bronze Age in northern Italy, plus some other older samples that are from related sites. And what we decided to do is we are really interested in some genes, in some part of some genes that are connected to this non-metric dental trait. that we wanted to be really sure to have enough of those parts of the genome, but still staying inside the economical budget that we have. So we decided to ask to Twist Bioscience to create a new custom spiking to add to their 1.4 million base panel. So to have to cover the part of the genome of the genes related to the specific question of this new project. So what I wanted to say is that not only you can do a lot of things with the Twist Bioscience capture, but you can also try to answer more questions and ask how to increase the number of SNPs covered without any problem. Maybe I spoke too fast, but I want to thank you, especially the Twist Bioscience in general for the opportunity, and especially Vishal Dixit and Gemma Garcia and Ian Hastings that helped me, that invited me and helped me with this presentation. Of course, all the other partners that are involved in the research, the group of minds, and my colleagues from Ferrara.